Homeostasis maintains optimal conditions for enzyme action and cell function, and it controls blood glucose concentration, body temperature, and water levels. Automatic control systems include the nervous system and the endocrine system. A receptor is a type of cell which detects a stimulus, and this is a change in the environment. Coordination centers include the brain, the pancreas, and the spinal cord, and effectors include muscles which contract and glands that release hormones. Electrical impulses travel along neurons. They're long, branched, and insulated. The central nervous system includes the brain and spinal cord. A reflex arc shows the pathway of electrical impulse, from stimulus to receptor to sensory neuron to relay neuron to motor neuron, and then to an effector, either a muscle or a gland. These responses are rapid, automatic, and they don't go through the conscious part of the brain. They are there to protect us from harm. Cerebral cortex, responsible for conscious thought and memory, the cerebellum, responsible for movement and balance, and the medulla, responsible for breathing and heart rate or unconscious activities. For higher tier students, you need to know that the MRI, scans, and electrical stimulation are used to study patient brain activity and the different regions of the brain. There are risks with brain surgery, such as infection and damage to delicate tissues. Retina at the back of the eye contains light receptors called rods and cones. The optic nerve allows electrical impulses moving from the eye to the brain to help us perceive images. The sclera is a tough white layer that protects the eye. The cornea refracts light as it enters the eye. The iris controls how much light enters and has a color to it. Ciliary muscles and suspensory ligaments help with accommodation, which we're going to talk about next. Accommodation allows us to focus on near and far objects. Let's talk about how this is done, as the lens actually changes shape to do this. So to focus on near objects, ciliary muscles contract, suspensory ligaments loosen, and the lens thickens, which allows strong refraction of light. Focusing on distant objects, however, the ciliary muscles relax, suspensory ligaments tighten, the lens becomes thinner, and there is a weaker refraction of light. There are two conditions you need to know about. Myopia is short-sightedness, hyperopia is long-sightedness, and corrective lenses in glasses or surgery can actually correct both these issues. The eye responds to different stimuli in terms of light. In bright light, the pupil contracts so less light can get into the eye. The circular iris muscles contract and the radial muscles relax, allowing this to happen. In dim light, pupils actually relax, meaning the circular iris muscles relax and the radial muscles contract. The brain contains a thermoregulatory center in the hypothalamus. Receptors detect the temperature of the blood, and receptors on the skin also coordinate the response. If we get too hot, vasodilation and sweating occurs, and as the sweat evaporates, it takes away thermal energy from the body, cooling us down. If we get too cold, however, vasoconstriction occurs and our body begins to shiver. This contraction of skeletal muscle actually releases heat energy, warming us up. And this is an example of negative feedback. The endocrine system is made up of glands that release chemical hormones into the blood, which act on target tissues and cells. They have a slower but longer lasting response than the nervous system. You need to know the following glands and hormones. The pituitary gland, or the master gland, releases FSH, LH, and ADH. The pancreas releases insulin and glucagon. The thyroid gland releases thyroxine. The adrenal gland releases adrenaline. The ovaries release estrogen and progesterone, and the testes release testosterone. Let's talk about glucoregulation. Blood glucose concentration is controlled by the pancreas. If it becomes too high, the pancreas produces the hormone insulin, and glucose moves out of the blood and is stored as glycogen in the liver and in muscle cells. If the blood glucose becomes too low, the pancreas produces glucagon. Glycogen is then broken down into glucose, and that glucose then re-enters the bloodstream. Diabetes type 1 is when there's not enough insulin produced, and therefore a person would need to inject insulin. Diabetes type 2 is when the cells don't respond to insulin anymore, so there needs to be more exercise and less carbohydrates in the diet. Back to triple content, feel free to skip ahead. This is osmoregulation. Water is lost in the lungs when we exhale, and also water, ions, urea is lost from the skin when we sweat, and urine from the kidneys filtering the blood. Remember, osmosis is a key principle here, and because the animal cells have no cell walls, if a cell gains or loses too much water, it can cause dysfunction. The kidneys produce urine by filtration of the blood and selective reabsorption of useful substances such as glucose, some ions, and water. For high tier triple students, you need to know that digested proteins actually break down to amino acids, and excess amino acids need to be deaminated in the liver and produce a toxic chemical called ammonia. Ammonia needs to be broken down into urea so it can be safely stored and excreted when it's in the body. ADH is a hormone that is released by the pituitary gland and acts on the kidney tubules to control water levels. Blood is too concentrated and therefore needs more water. ADH is released, so more water is reabsorbed into the blood from the kidney tubules, and this is an example of negative feedback. If the kidney no longer works, there are two options. You either have organ transplant or dialysis. Organ transplant essentially removes the damaged kidney and replaces it with a donor kidney, meaning the organ now functions correctly, and there is no real need for dialysis. However, there can be long waiting times until you get the correct correct donor and you get a genetic match. However, you also need to be aware that there are surgery risks, death, infection. Also, immunosuppressant medicines, which reduce the chance of rejection, will be needed, but this can lead to opportunistic disease. Dialysis is an artificial method that we use to filter the blood of kidney failure patients. It can be expensive, but it reduces the urea in the person's blood, and there's no change to glucose concentrations here. There are also other negatives where you need to do this several times a week for hours at a time, so this can be life-altering. Unfiltered blood of a kidney failure patient is actually high in urea and is mixed with anticoagulant or blood thinners to prevent clotting as it enters the dialysis machine from the patient's arm. Once inside the machine, the blood and dialysis fluid are separated by a partially permeable membrane. The blood flows in one direction and the dialysis fluid actually flows in the other, and the dialysis fluid contains glucose, ions, and no urea, and the exchange actually occurs between the concentration gradient. Puberty is a sequence of events that occurs at a set time in a person's life, coordinated by hormones, and it causes secondary sexual characteristics. In female people, this is hormones including estrogen, which thickens the uterus lining, progesterone, which maintains the uterus lining, FSH, which matures the egg cell, and LH, that spikes at day 14 and releases an egg cell during ovulation. In male people, this is testosterone, and the testes produce testosterone to help with sperm production. For higher tier students, you need to know that progesterone and estrogen inhibit LH and FSH, so no more eggs can be released and mature, and this is an example of negative feedback. Different methods to prevent pregnancy. First are hormonal methods that inhibit FSH and LH, including the contraceptive pill or the patch, the implant, the injection that contain progesterone, and the intrauterine device or IUD that also prevents implantation. Non-hormonal methods include barrier methods like condoms and diaphragms, another type of IUD which is non-hormonal that prevents implantation, spermicidal agents that kill the sperm, abstaining, and surgical sterilization. Let's talk about infertility, a higher tier topic. Infertility is the inability to reproduce. Steps involved in the treatment using IVF. FSH and LH are a fertility drug that's injected. The egg cells are collected and fertilized by the sperm in vitro or in glass or in a laboratory. The fertilized eggs develop into embryos, and then they are divided to become tiny balls of cells. One or two of these embryos is then inserted back into the uterus. There are limitations, including emotional, physical intensity, so this can affect someone's mental health. There are low success rates and also multiple pregnancy risks, and this can affect the baby and the mother as well. Negative feedback for higher tier students. You need to know that adrenaline and thyroxine are two hormones that undergo negative feedback. Let's talk about adrenaline first. It's produced by the adrenal glands, and this is during times of stress or fear. As a result, increased heart rate, more oxygen and glucose moves to the brain and muscles, preparing for a fight or flight response. In terms of thyroxine, it's produced by the thyroid gland, and it stimulates the basal metabolic rate, and this is for growth and development. Another triple topic, feel free to skip ahead if this doesn't apply to you. Plant hormones. So phototropism and geotropism are two ways plants respond. Phototropism is responding to light by hormones, and geotropism is responding to gravity by hormones. The hormone is auxin, and it's made in the root and shoot tips. Auxin can be concentrated by diffusion on the shaded side, causing more cell elongation. It also inhibits the growth in roots, so the roots actually move downwards towards the ground. Higher tier students only need to know the different uses of other hormones. So gibberellins helps end seed dormancy and increase fruit size. Ethene is used for fruit ripening and cell division, and auxin can also be used for weed killers, rooting powder, and growth that affect human reaction time, sometimes called the ruler drop test. Let's talk about the method. Person A will sit down and put their hand out, resting their arm on the table, and have a gap between their thumb and forefinger. Person B will hold a ruler vertically above person A's hand with the 0 centimeters facing downwards. Person B will drop the ruler for person A to catch without telling person A. They then will record the reading above the thumb and repeat 10 times and calculate the mean. Now they need to repeat this with the factor being investigated and convert all readings into time using a conversion table. The result should be a faster reaction time or less time if the person is not distracted, or if they have a factor that increases reaction time. So for example, you could have noise levels, which could actually increase reaction time, or you could have caffeine that is more likely to reduce reaction time. Lack of sleep as well could increase reaction time. You also need to know about the different variables such as control and dependent. So the dependent variable is the centimeter length reading, and the controls include the height of the ruler, the person catching. Let's look at a required practical for triple students only. This is for germinating seeds and looking at tropisms. So the first thing you want to do is add 10 seeds to a Petri dish, 3 in total, containing cotton wool and the same volume of water, and allow them to germinate for a few days. After this, you want to record the height of the seedlings. Once grown, place the Petri dishes in either A, full sun, B, in a dark cupboard, and C, in partial light, maybe a cardboard box with part of it cut out. After 7 days, you want to record the new height and calculate the mean. So the results, the more light there is, the more growth. Shoot growth at an angle, maybe towards the light light is a phototropism. If the seedlings with no light, this is often going to cause the most growth, but they're going to be very spindly and yellow. So the independent variable is light intensity, because that's what we're looking to investigate. The dependent variable is the length, and the control is the seed species and the time of the growth period, and maybe water volume as well. Next topic, you guys, inheritance, variation, and evolution. Sexual reproduction includes meiosis as cell division, leading to genetics being mixed together, causing variation. As a result, you have gametes, which are haploid, so the sperm and egg cell. In asexual reproduction, only one parent is needed. There's no fusion of gametes. This is including mitosis as a cell division type. So clones are produced, they are identical genetically speaking, and no gametes. These are diploid cells, these are somatic cells. Benefits of asexual reproduction include only one parent is needed. It's more efficient because you don't need to spend time or energy finding a mate. It's therefore faster and many clones can be produced. However, sexual reproduction allows for variation. You also have more survival opportunities as the environment changes, and selective breeding is a result of sexual reproduction. Organisms which actually use both methods include malarial parasites, fungi, and strawberries and daffodil plants. Gametes are produced by meiosis, a type of cell division, the sperm cell in the testes, the egg cells in the ovaries. The steps include genetic information, which is copied, 2 cellular divisions, 4 daughter cells are produced, leading to haploid varied cells. The sperm and egg cell can then fuse together in terms of their nuclei and form diploid cells by mitosis. DNA is a polymer made of 2 strands and in the shape of a double helix. It's contained in chromosomes in the nucleus. A gene is a small section of DNA that codes for amino acids that make proteins. The genome is the whole sequence of DNA of an organism, and the human genome was mapped, which has been helpful for medicines, treatments of diseases, inherited disorders, research, and ancestry. DNA structure for triple students only. The nucleotide is the monomer of DNA. It's made of phosphate, deoxyribose sugar, and bases A, C, T, and G. 3 bases equals 1 amino acid, and the order of amino acids determines the protein. Protein synthesis, triple students high tier only. Base pairing includes A binds to T, C binds to G. The order of amino acids determines the protein, and mutations can change this order and therefore the protein. Proteins are synthesized by ribosomes, and mRNA is a copy of DNA, which allows this process to happen. Protein folding is determined by the order of amino acids, so the folding links to the function of the protein, such as collagen. If non-coding parts of DNA are mutated, this does not code for a protein, and therefore it won't affect the protein. Key terms you need to know. A gamete is a sperm and egg cell, or the sex cells. Chromosomes are thread-like structures made of DNA. A gene is a small section of DNA. An allele is a version of a gene. Dominant alleles are always expressed. You only need one. Recessive alleles are only expressed if 2 alleles are present. Homozygous means 2 of the same allele. Heterozygous means 2 different alleles. Genotype is a combination of alleles, and a phenotype is an observable characteristic. Genetic diagrams are used to interpret and predict ratios or probability of diseases or traits that are passed on in family trees. First, you could use a Punnett square, so complete the diagram, or higher tier students, you could be expected to construct them. Family trees show multiple generations and relationship between disease carriers and non-affected individuals. Polydactyly. This is when a person has extra fingers or toes, and it's caused by a dominant allele. Cystic fibrosis is a genetic disorder where it affects cell membranes, and it's caused by a recessive allele. In human diploid somatic cells, there are 23 pairs of chromosomes, or 46 in total. 22 of those pairs control characteristics, and one of those pairs determines genetic sex. XX chromosomes shows as a female, XY chromosome shows as a male. You could be expected to complete or construct Punnett squares, but you need to know that there's always a 50% chance of a female and a 50% chance of a male, even with multiple pregnancies. Variation is differences in the characteristics of individuals in a population due to either genetics that are inherited or the environmental conditions, or both. All variation comes from mutations, which is random and continuous, and it's very rare for new phenotypes, but it can be advantageous if there are mutations that can actually change a species. There are theories of evolution, one being natural selection you need to know about. It states that all life came from simpler forms around 3 billion years ago due to mutations that arose within the population, leading to variation. This meant the best adapted survived, passed on their genes through reproduction, and this continued through multiple generations, leading to different species. A species is one that can interbreed to produce fertile offspring. Darwin came up with this idea of natural selection, and he wrote his theory in the book Origin of Species in 1859. He stated that variation happens between species, the best adapted survive, and they pass on their genes to the next generation. There was controversy at the time because it challenged the idea of God, and there was a lack of evidence. Other theories like Lamarck's stated that changes occur within an organism's lifetime. Selective breeding is a man-made technique used to create disease-resistant crops, organisms with more meat, egg, or milk yield, domesticate animals, or for aesthetic reasons. The steps include desired characteristics are identified in the parent species. The parent species is bred together, and the offspring are selected with the desired traits. These are then bred together and repeated over many generations. However, this can lead to inbreeding, which can result in disease or genetic disorders. Genetic engineering is a key idea introducing genes from one species into another to get the desired characteristic, for example, the human insulin gene and disease-resistant crops. The steps include the desired gene is isolated and cut out via enzymes. The gene is inserted into a vector of a plasmid, which is found in bacteria, using the same enzyme to cut the plasmid's DNA. The recombinant gene is inserted into the bacteria, and this divides by binary fission. The gene is then inserted into the plant or animal during early life to allow it to be incorporated into their genome. Culture is an example of cloning, and this is used to create small parts of a plant, especially rare species. You can also use cuttings from an established plant, or embryo transplant, which is splitting up the embryo of an organism. Adult cell cloning is a key example you need to know about. So first, the nucleus of an unfertilized egg is removed. The nucleus of a somatic cell is then inserted into the egg cell. An electric shock is used to stimulate growth, and the embryo divides and is inserted into the uterus. This results in the same genetic or a clone of the somatic cell donor. Alfred Wallace also suggested Darwin's theory of evolution. He was also really well known for his work on warning colours in animals and speciation. The steps in speciation include isolation. A geographical barrier separates a population. A mutation occurs in both populations. Natural selection occurs with the steps we've discussed, and then divergence. So the genetics vary so much that each population can no longer reproduce. So they are no longer able to produce fertile offspring. So they are their own separate species. He did a lot of observations with pea plants and inheritance experiments. His observations led to this idea of units that were passed down unchanged to offspring. After his death, there was the discovery of DNA structure, and this led to this idea of units being called genes. Darwin's theory of evolution, natural selection, is now widely accepted due to fossil records and bacterial resistance. Fossils are remains of organisms or traces in burrows, footprints, rootlets, and bones. And this is when, over millions of years, soft tissues of those organisms decayed and they were hardened in mineralization and are preserved. Fossil records are actually incomplete because not all of them are found. Some have been damaged, and soft-bodied organisms have decayed without a trace. Extinction is when no remaining individuals of a species are still alive. There are factors that lead to extinction, including new predators, disease, environmental disasters like drought, fire and flood, asteroid collision, and inter- and intra-species competition for resources such as food. Because bacteria can reproduce rapidly, we can actually see that evolution quite quickly. Mutations within a population give rise to new strains. New strains have antibiotic resistance genes and therefore are not killed by antibiotics. They survive, reproduce, and pass on their genes. A key example is MRSA. How this is done is because of the misuse of antibiotics and the overuse of it, with people having viral infections being prescribed this treatment. Also, it's used in agriculture and people not finishing their course. Organisms have been classified based on observable characteristics by Carl Linnaeus. His system included the kingdom, phylum, class, order, family, genus, species. A binomial name or an organism's Latin name is based on the genus and species. Due to new technology in microscopes and biochemistry, classification systems have developed, with Carl Woeser's three-domain system, which has been added to Linnaeus' system. This includes archaea, bacteria, and eukaryota, and evolutionary trees show the relationships of life. Last topic, you guys, let's talk about communities. This is all populations of different species living in a habitat. A stable community of factors that are balanced so populations remain fairly constant. Interdependence tells us that species rely on each other for food, shelter, pollination, and seed dispersal. If one species is removed, it can affect the whole community, and you see this in predator-prey cycles. Biotic factors are living factors such as food, predators, disease, competition, and territory. Abiotic factors are non-living, like light intensity, temperature, water or moisture levels, soil pH, mineral content, wind intensity and direction, carbon dioxide levels, and oxygen levels. Adaptations help organisms survive in their environment. Adaptation types include structural, such as fur, blubber, large surface area, and camouflage, behavioral adaptations like migration or nocturnal patterns, and functional adaptations like concentrated urine to reduce water loss. Extremophiles, such as bacteria or organisms that live in extreme environments like hot springs and volcanoes. Levels of organization: organism, population, community, and ecosystem. An ecosystem is the interactions between living and non-living parts of the environment. Cycles include the carbon cycle, where there are processes like respiration, decay, photosynthesis, and combustion, where carbon is actually cycled throughout. The water cycle is also important because this is where water is cycled in the environment during evaporation, precipitation, and condensation. Decomposers play an important role in material cycling because they return mineral ions to the soil, so worms, insects, bacteria all play their part. This is the breaking down of dead matter. Microorganisms involved are bacteria and fungi, and you need warm, moist, and oxygen or aerobic conditions for this to thrive. And often you are asked about compost as a key example. Biogas generators use anaerobic conditions and methane gas is produced. Changes from deforestation, global warming, and pollution can change our environment and therefore species. Slower changes like seasonal changes, new predators or pathogens can also have an impact. Indicator species tell us of that impact. So changes in population indicate pollution levels, such as lichens indicate air pollution, and invertebrates indicate water pollution. Another example of this is the peppered moth. Biodiversity is a variety of different different species in an ecosystem. High biodiversity is found in stable ecosystems and less affected by environmental change. Humans can actually cause an impact at reducing biodiversity by deforestation, habitat destruction, pollution, and global warming. Waste disposal methods include landfill use. This is using a lot of land for landfills, which is a negative. Also, methane can be produced, and also toxic chemicals can leak out of landfills. Another method for waste disposal is incineration, which reduces land use. It can also be used to generate electricity, but toxic gases can also be released by burning waste. Another method is recycling, which is reducing landfill use and less land that's needed, but it does require energy to reform those substances. Humans use land for building, quarrying, farming, and waste disposal. Peat bogs are important carbon stores. Destroying peat bogs releases carbon dioxide, and peat can be used as a fuel and for compost. Effects of deforestation include loss of biodiversity, increased carbon dioxide as less carbon dioxide is removed by photosynthesis, and habitat destruction. Tropical rainforests have high biodiversity and are important for maintaining atmospheric gases. Global warming causes are increased greenhouse gases, carbon dioxide, and methane. The effects are climate change, rising sea levels, less biodiversity, and habitat loss. Methane is produced by cattle, rice paddy fields, and landfill sites, and carbon dioxide is produced by combustion of fossil fuels. Programs that maintain biodiversity include breeding programs for endangered species, protecting habitats, reintroducing field margins and hedgerows, reducing deforestation and carbon emissions, and recycling resources. Roles of scientists are monitoring species and protecting rare habitats. Feeding position in a food chain is the idea of a trophic level. Trophic levels, you have producers, plants and algae. You then have your primary consumers, which are herbivores, secondary consumers, and then tertiary consumers. Remember, biomass is the total mass of living material. Biomass can actually decrease at each trophic level, and pyramids of biomass are always upright and tapered in a pyramid shape. Biomass is transferred less efficiently because not all the food is eaten. Energy is transferred in respiration, and this is transferred as heat usually, and waste products are produced such as feces. We can calculate the transfer of biomass using an efficiency equation. Efficiency is equal to biomass transferred divided by biomass available times 100, and we can actually improve efficiency in farming by reducing the movement of organisms, so battery farming, keeping animals warm by using heating, and controlling the type of food that's being eaten by the organisms. Food security, triple science only. The next few topics are triple science, so feel free to skip ahead to the required practicals. Food security is having enough food for a growing population. Problems affecting food security include increased population, changing diets, new pests or pathogens, environmental changes, and cost of agricultural inputs. There are two main types of farming methods, the first being intensive farming. They are more efficient because there is a higher yield of organisms produced. However, there are ethical concerns, there are waste production concerns and disease concerns. Another method of farming is sustainable farming methods, which reduce energy and water use. They reduce chemical use. However, there is a lower yield. Sustainable fisheries. So problems with overfishing cause fish stocks to decrease, biodiversity to be reduced. Sustainable fishing methods include net size limits, fishing quotas, and protecting breeding grounds. Biotechnology is another method of creating food, and it uses microorganisms to make biofuels and medicines as well. Mycoprotein is a protein-rich food made by a fungus and is grown aerobically in fermenters. Slow-moving organisms and plants, we can calculate the abundance and distribution using either a quadrat or a transect. A quadrat is a square frame that's used to measure percentage cover or abundance, and a transect is a quadrat and a measuring tape. The method to use for each, a quadrat you want to make sure is placed at random coordinates that avoids bias, whereas a transect you want to place the quadrat at regular set intervals. In both cases, you want to calculate the mean average. Remember, with a transect, you're looking at the effects of abiotic factors like light intensity, temperature, and soil pH. Practical is for triple scientists. It's investigating the factors and their influence on the rate of decay. So temperature, oxygen, and water or moisture levels. You use milk, lipase, and an indicator and measure the time for colour change. The faster the colour change, the faster the rate of decay. The key idea is that optimal conditions will increase the rate of decay. So for example, higher temperatures increase enzyme activity and therefore increase the rate of decay. Independent variables are one of the factors above. The dependent variable is the rate of decay all the time, and the control is any of the factors that aren't used, the milk type that you use, the volume of milk as well. I've been the GCSE science teacher and you have been curious. If you enjoyed this video, feel free to give it a thumbs up. Feel free to subscribe and join our amazing community. And if you like this video, maybe you'll like these ones as well. Also, good luck for your next exam and I'll catch you in the next one.